Hi, my name is Chase Stevens, and I'm a fifth-year pharmacology and toxicology graduate student in the Phen Lab at UC Davis. My research on ozone helps asthmatic patients who don't currently respond to existing therapies by identifying additional targets for drugs used to treat asthma. The air we breathe contains a multitude of air pollutants that pose a risk to human health and worsen or possibly cause lung disease. Ozone and other air pollutants are a particular concern in California and nationwide due to the large percentage of the population exposed to them. My research seeks to address the mechanisms by which these pollutants can worsen existing cases of asthma using a mouse model that recapitulates key characteristics of the disease. The mouse model I'm using consists of allergic sensitization to house dust mite allergen, which is one of the most common allergens in humans. The sensitization process causes the muscles that constrict the airways to become more sensitive to subsequent exposures to either the allergen or to ozone, which mimics the hypersensitive airways of asthmatics. Subsequent exposures lead to inflammation of the airways and excessive production of mucus, which ultimately leads to obstruction of the airways and less oxygen reaching the bloodstream. Overall, my mouse model provides a realistic example of how humans are exposed to ozone and enables the study of how this exposure leads to characteristics that are typically observed in asthmatic patients. I utilize a variety of techniques to better understand the effects of ozone within the lung. Many of these techniques revolve around the use of metabolomics, which is the large-scale study of metabolites produced within an organism. Metabolites are small chemicals that are constantly produced within our bodies as a result of food digestion, maintenance of our cells, and exposure to chemicals in our environment. Importantly, metabolites can also be produced to carry out important signaling functions or communication between cells. Metabolomics can simultaneously measure the quantity of thousands of metabolites in a single experiment, which provides a snapshot of metabolism related to biological function and is influenced by disease or environmental exposure. These measurements are routinely collected using mass spectrometry, which is suitable for accurately identifying and distinguishing this large number of metabolites in a sample containing cells, tissue, or fluids such as blood from an organism. My project also incorporates mass spectrometry imaging to not only measure the composition of metabolites within the lung, but where these metabolites are located. Overall, this information is essential to improve our understanding of the effects of ozone exposure and to develop additional drugs for the treatment of asthma in populations that don't currently respond to available treatments.